Hello everybody, my name is Shelby Harris and I'll be presenting to you on gene editing by the use of CRISPR-Cas9 on epigenetic factors such as DNA methylation. So to start off, what is epigenetics? Epigenetics are inheritable changes in the DNA that occur due to environmental factors. This most commonly happens by either DNA methylation or demethylation. A great example of this is the Dutch Hunger Winter, in which the whole area of the Netherlands really went through a famine where the adults were starving, and when those adults had kids that during that winter, they in earlier life when they did have food, they were very, very prone to being obese. This is because certain parts of their DNA had been changed and altered due to that environment of where their parents were starving to where their DNA said, well, oh, we're going to upregulate these genes that keep fat on our bodies so that when we go into that world and we have to live in it, we can easily survive because they're being able to keep on fat. But when that famine ended, those children became obese. And that's a great example of DNA methylation and how it changed a whole generation of Dutch children. And next we have, what is methylation? Methylation is the addition of a methyl group to a DNA base. This usually happens to cytosine and is caused by the enzyme DNA methyltransferase. This is done so that a promoter of a gene will get blocked by these methyl groups, and these transcription factors that usually transcribe the gene can't reach the gene, and it won't be able to transcript and become expressed as a protein in the cell because it can't be the transcriptase can't read it. DNA demethylation is the opposite of methylation, and this is where the methyl groups are removed, and then that gene is then upregulated because those transcription factors can easily get to it, and they can easily up the protein expression. DNA methylation has been found to have roles in many different diseases and illnesses, such as cancer, autoimmune diseases like arthritis, lupus, MS, diabetes, and even some neurological disorders like Rett syndrome. It's also been found to have extremely important roles in embryonic development because there are certain genes that need to be upregulated and downregulated all throughout fetal development, or things go horribly wrong to your fetus. So next we have a CRISPR-Cas9 review. So of course CRISPR-Cas9 is that big, huge, crazy genome editing tool that we use. Everybody talks about it all the time. It's very popular. CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats, and those are essentially areas of the DNA of the bacterial immune system that will be palindromic and they repeat, that were essentially segments of viral DNA that the bacteria kept back so that it could put into the CRISPR-Cas9 guide RNA, and when it gets infected with a virus, it will scan all the DNA, and if it finds viral DNA, it will chop it up and essentially, inactivate the viral DNA so the virus can infect the bacteria anymore. So like I just said, it's a bacterial immune system. How it works is that the Cas9 enzyme, which is this enzyme right here, essentially scans the DNA and compares it to its guide RNA. And when it encounters a sequence that matches, it cuts the DNA on both sides. It does a double-stranded cut, and this has been used in the lab, harnessed in the lab, to where if you put in a special segment into your guide RNA, it will cut where you want it, and then you can further add in DNA sequences. Here, if you want to, you can add in stuff, or you can remove stuff. You could remove this entire genome section, which is essentially what they're trying to do with, say, like cystic fibrosis. That's a single gene mutation. They're going to try to go in there with CRISPR-Cas9 and cut out that gene and put in a new one in that functions correctly, so that those people don't have CF anymore. Why is it so good of a DNA editing method is that it's very, very accurate and is actually very, very easy to use. It's very easy to make these enzymes. You can do a lot with them, and they have little no off-stream effects. They rarely ever go wrong, so they're extremely accurate and they're easy to use, so they just make it all around a great tool to use in your studies. So examples of some work that I found of using Cas9 on DNA methylation was one work from 2016. It was reprogrammable Cas9-based system for inducing site-specific DNA methylation. James McDonald's in his lab found out how they can develop a Cas9 transfer methylase that will cause DNA methylation at specific sites in the genome by incorporating that transmethylase into the Cas9 and deactivating the nuclease activity of the enzyme. So essentially what they did was they took the Cas9 enzyme and they gave it transmethylase enzyme activity, and they took out the endonuclease activity so that it couldn't cut, but it could add on methyl groups. And my next work that I found was also in 2016, and it was editing the DNA methylation in the Mendelian genome. And this was very interesting because not only did he prove that he could methylate certain genes, but he also found that he could do the inverse and demethylate certain genes using another modified Cas9 enzyme. So that was really cool. And my examples continue. So these are actually both from 2020, brand new. This top one here, editing DNA methylation in Mendelian embryos, was just done in January. It was just published in January. And this doctor, what he did was he used methylation editing in germline cells in mice to show that methylation is very, very important for development. He found that if you knock out the genes for methylation, or say if you methylate a very, very important gene that's not supposed to be methylated at that certain time, he found that it really messes up the mice embryo, that they do not develop correctly, and they typically die prematurely due to the fact that they don't have these transferases, or their genes are methylated in the wrong areas at the wrong time. The second work was DNA methylation editing by a CRISPR-guided exclusive of the 5-methylene cytosome. So this researcher, what they found was that this plant enzyme, DCAS-ROS1, it can demethylate genes without having to replicate the DNA, which is something that the human demethylating enzyme has to do. So since this plant enzyme doesn't have to replicate the DNA, they were hoping that they will be able to modify it and use it in the human editing of the genome, so that we don't have to go through those extra steps that we currently have to go through when we're using the human demethyltransferase. So to end off, I'll teach you about the future of gene editing. Gene editing is changing the game and how we view disease treatment. Essentially, back in the 1900s, if you had a genetic disease, that's just it. You could treat the symptoms, but there's really nothing you can do about it in the end. But we're now finding out that we can use these gene editing systems to edit the genome and change it up so that we can try to fix these diseases that typically were incurable before now. So that's going to be a really huge benefit of using gene editing in medicine. It's really easy to use and really accurate, making it a great tool to use. It's being used in a lot of different areas. Obviously, if you look at this picture over here, it's being used in fuel, food, materials, rapid genome studies. So like for our mice and stuff like that, of course, we did the rapid embryo study with mice that teaches that methylation is really important for development. So gene regulation, of course, we have anything that would also tell us like how the genes work. Drug development, we can make, that's how insulin's actually made, was using edited E. coli to essentially produce human insulin for easy use, because we used to have to use pig insulin and sometimes people would have allergic reactions to it, and that would be really bad because then they ran out of the one way they could get insulin and they would then die of their diabetes, which is not a great thing. But now since we have that GMO E. coli producing human insulin that will not have an allergic reaction to if you use it, that really saved a lot of our diabetics. And then, of course, gene therapy, like I said, with the cystic fibrosis, Huntington's disease, and other diseases that are single mutations like that, they're probably going to be in gene therapy trials here really soon. And more advanced diseases that probably has a lot of different genes going into their development and their prognosis will probably be looked at here in the next century. It probably won't be that long before we get to those. But even though we still have all these benefits, there's still fears around this practice. Of course, everybody's scared of GMO food. Certain people don't eat it just because they're scared of it, or they don't know, or they don't like it. It's such a new frontier in science. We've never have to ask these questions. We've never seen anything like this. So we can't really say what's going to happen, which is always scary. You always want to know the answer, but even though you don't know it, it's really nerve wracking of what could possibly happen. But I think gene editing will become a really reality really here soon. It's too the benefits we get from gene editing is way too strong to just throw it out the window and say we're too scared to do it. I definitely feel like we're gonna start using gene editing in modern day medicine, just normal everyday medicine, really, to treat a lot of different things. And I feel like it's going to become a really, really strong part of how we treat diseases in the future. Well, anyways, here are my work sites. Thank you for watching my presentation.